Hello, and welcome to my poster here at CSEP 2020. Thanks so much for coming. I'm going to go ahead and zoom in here, and we're going to work our way through, beginning with the introduction. So, in my prior research and the work of others, it was demonstrated that plasticity in the human motor cortex is enhanced immediately following a bout of cardiorespiratory exercise. Here, we performed a retrospective candidate gene analysis to explore potential sources of inter-individual variability in the data. We hypothesized that people genetically predisposed for low dopamine D2 receptor availability, so carriers of the Lyse allele of the DRD2 ANKK1 polymorphism, and people predisposed for low activity-dependent secretion of brain-derived neurotrophic factor, so the carriers of the MET allele of the BDNF Val66 MET polymorphism, would show the greatest exercise-induced increase in motor cortex plasticity. So scrolling down here, figure 1 provides a general overview of the experimental design. Focusing in on this right side of the figure, you can see that all participants completed 2 experimental sessions in a randomized order. Depending on the session, participants either performed high-intensity cycling intervals or a period of seated rest. Immediately before, we used a transcranial magnetic stimulation, or TMS technique, called paired associative stimulation, or PAS, to induce neuroplasticity in the motor cortex. A larger increase in corticospinal excitability following PAS indicates greater motor cortex plasticity. So jumping right over to table 1, this is where you can find the general characteristics of our participants. You can see that our total sample was 16 young healthy adults. And figures 2 and 3 here depict the results shown in our prior work, indicating that when preceded by exercise, PAS had a larger effect on corticospinal excitability. So you can see that sort of raw MEP input-output data here, and over here you can see the group effect, and then broken down by individuals from the rest and exercise conditions. So moving over to figures 4, 5, and 6 is where you can find the new results presented in this poster. And what we've done here is break down those prior findings based on the genotypes of interest. And so what you find here is on the y-axis, we're showing the percent change in corticospinal excitability elicited by PAS. And figure 4 depicts a statistically significant interaction indicating that response to PAS was facilitated by exercise for DRD2 Lyse allele carriers, those ones in green, only. If you look over in panel B, you can see the results across each individual. When we move down to figure 5, what you're seeing is those same results, but instead broken down by the BDNF genotype. And here, the results are much less clear. All right, there's a lot more variability and no clear impact of the BDNF polymorphism on response to exercise. As we move down to figure 6, we also considered the potential impact of having both the DRD2 Lyse allele and the BDNF MET allele. And what we saw are some results that suggest that potentially those individuals who carry both of those alleles are in fact the ones that experience the greatest exercise effect. However, there is a lot of variability in this data and few participants, and you can see that the effect is driven particularly by these 2 participants. So overall, these findings suggest a potential role of the dopamine D2 receptor in mediating acute exercise effects on human motor cortex plasticity. Nevertheless, further work is needed to increase sample size and consider the effects of other variables, from variables like cardiorespiratory fitness level right through to other genetic variants. Thanks so much for your time. I'm happy to answer questions in the chat. Bye now.